Just wanted to remind everyone that the college has been sending out communications regarding what's going on with the vaccines and things like that. And so the plan is to return to in-person classes. Sorry, I'm just getting things organized here for the class, letting people in. Plan is to return to in-person classes October 4th. So I'm not here to answer all your questions on how all that's being implemented. Just make sure you do read your emails. If you have questions, you can reach out to the department chair or the college nurse, and always check your emails and look at the college website. There's a link there you can check out. So on with today. Last day I had finished off on this particular slide here, talking about lab reports. And I'd mentioned I would just talk a little bit about titles next day. And so here we are, it's next day. So a little bit more on titles. You are going to have titles for your report. So you have to give your report a title. And you're also going to have to give any of your graphs in that report titles. And this will be something you'll be doing all semester long. Every time you make a graph or a table, it's going to require a proper descriptive title. So here are some tips, and I think we mentioned this before, is that you're going to have in most of your titles, you're going to have the relevant binomial name. So this is the binomial name right here for beets, so it's beta vulgaris. Titles need to be informative. Say what was done. So don't just tell me that it's something like distance versus time or temperature versus concentration. That's just reading off the axes. Give me some information about what was done in the experiment and sometimes even why. So I have a couple of examples here, right? You can see first one, effects of cold temperature on humans. Not too bad, maybe a little bit basic. Maybe in some circumstances that might be an okay title. But you can see, take a look at the second one there. Notice it has the binomial name of the species. And tells me a little bit about how this was done. If you need to make your title into, let's say, 2 or 3 sentences, you can do that. And in some cases, you'll see that's the case in scientific papers. Some people go a little nuts. You don't need to go nuts here, but what you do need to do is be relatively descriptive in your title, and do not forget your binomial name. The other thing to not forget, of course, for your figures is all your figures are going to be labeled figure something, and this could be figure 1. That's supposed to be a 1, or figure 2, or figure 3, or something like that. If you have any questions, let me know. On Wednesday, I'm going to talk about some of the writing parts, the introductions, and maybe the methods. Back to topic 4 today. Last time we were talking about cell membranes and cell membrane transport. Today we want to talk about cell walls. So cell walls are always exciting. That's what I did some of my PhD research on, and the bacterial cell wall in particular. So let's just think about membranes and cell walls, and then there's that third term there, extracellular matrix. I kind of want to just define these things before we get any further. So remember, this cell membrane is a phospholipid bilayer. It's going to have proteins, and it encloses all cells, and it allows things in and out, and has a whole bunch of different functions. So what's a cell wall? A cell wall is something found outside of the membrane, and it's not fluid. In fact, it's a lot more rigid, and it's not found on all cells. Okay, and we'll take a look at some cell wall types today. An extracellular matrix is something that's found in animal cells, and it's not technically a cell wall. It can be rigid. Sometimes it's actually very non-rigid. It's very floppy or flexible, and so it's kind of something not even found in all animal cells, in some animal cells. So I'll give you some examples of extracellular matrix in a few minutes when we get to that part of the lecture. So first, let's talk about the plant cell wall. This is probably the one that most people are familiar with. It gets talked about a lot in as kind of a basic plant cell type of thing. Way back is probably as far as grade 6 or something like that. Here's your typical plant cell, and you can see the cell wall there is in green. Cell walls aren't green, but plant cells are green. So you probably know that the chloroplasts are the part that makes it green. But in textbooks, they often color them green because plant cells are green, and green is a lot better than white because that's what the cell wall would be white. So I kind of want to zoom in on this, and this diagram thankfully does. And you can see what you're actually looking at is several plant cells there. So here, for example, this is plant cell 1, and then we have another cell down here, 2, and another cell over here, 3. And so that's the junction of the 3 cells. And there's a little bit of material in the middle here too. We'll talk about that in a minute. So by the way, this here is the cell membrane. And so you can see that the cell wall is outside of the cell membrane. So what's a plant cell wall made of? It's made out of cellulose. So go back to topic 2, we were talking about macromolecules, and I was mentioning with carbohydrates, you can link them together, and you can link them together in different ways. And if you link glucose together in one way, you get starch and glycogen, which is an energy carbohydrate. If you link glucose another way, you get something called cellulose. So notice here that the cellulose, each glucose monomer is basically upside down compared to one another. And I think I showed you this diagram here to kind of show you what's going on in 3 dimensions with cellulose. So what is going on is that when you link them in that particular formation, it's called a beta glycosidic linkage, you get all sorts of hydrogen bonds. So all of these little dotted lines here, there's a hydrogen bond there, and there, and there, and there. So a hydrogen bond, if I remember correctly, is about maybe a quarter of a strength of an ionic bond. So it's a weak molecular force. But in cellulose, you have hundreds, thousands of these hydrogen bonds. And even if you have a weak force, when you multiply that by hundreds and thousands, it actually makes a relatively strong structure. So that's cellulose. It's a glucose polymer, and it's relatively rigid and strong, and it's a tough material. So tough materials are good for, of course, cell walls. Here's the image from the textbook. Maybe not quite as nice, but you can see if you look very carefully, these lines here are dotted and representing hydrogen bonds. So these bundle together, and you make these things called, you get cellulose molecules, and they bundle together, and they're called cellulose fibers, or sometimes they're called microfibers or microfibrils. And these get together, and then you have the main part of the cell wall. So here's sort of another diagram of the cell wall, and you can see that the cellulose fibers are right here. These kind of bluish turquoise colored things there. You may notice there are other things in a cell wall. Cell wall is not just made out of cellulose. There are proteins in there. You may recognize this one here, pectin. I don't know if anyone knows where you might see pectin. Pectin is something that people use often to make jam or jelly or something like that. So you can buy it at the grocery store. It's just a protein, and it's kind of a, I guess you could call it a gellifying agent, right? That it's kind of sticky and sort of glues the whole thing together. So if this were concrete, if the cell wall was concrete, the pectin would be kind of like a soft cement, and the cellulose would be like rebar, if that makes any sense to anybody, if you know a little bit about concrete. So let's just take a little bit more careful look at the plant cell wall here. And what I'm going to do for you is kind of try to draw a little picture here and to explain what is going on. So if you take a look, hold on a second, let me just see here. Take a look. Yeah, okay, I have this all kind of I mean, to see where I have space on the slide. So the first thing to mention is we have a primary cell wall. So I'm going to draw a little picture over here, and I'll probably do a little bit of erasing as well. So first thing you're going to have is your plasma membrane. That's your PM. And then what happens is cellulose is made, and so you end up with your primary cell wall. So this is my primary cell wall. And so that's the very first thing made, and that's what you see in a lot of plants. So I mentioned I was going to do a little bit of erasing, because it says here, what happens is sometimes you get a secondary cell wall. So here I go with some erasing. I'm going to erase this here. No, I can't erase it. Why can't I erase it? Maybe I need to find the right tool. Wonderful. Pointer options and eraser. There we go. So what happens is, okay, now I've got to change my pointer tool back to a pen. So what happens is that primary cell wall actually gets pushed. So there's my primary cell wall. And then what you end up with is sometimes a secondary cell wall. There's my secondary cell wall. So the primary cell wall is made first, and it gets pushed outwards. And in many plants, you get a secondary cell wall, and the secondary cell wall is usually kind of woody. So this is where you get all sorts of different wood carbohydrates, such as xylans and things like that. You don't need to know what a xylan is and whatnot, but just know that some plants, particularly woody ones, do make a secondary cell wall, which pushes the primary cellulose cell wall a little bit further away from the cell membrane. You may also notice that between the cells here, you have this lamella, and that involves a lot of pectins. I told you pectins are kind of sticky, and it's basically gluing the cells together. So one more thing you may have noticed is down here, this label for something called plasmodemata. These are basically little channels between the cells. So of course, plants, they like water, and water has to be transported across the plant very quickly. So these are there so that water doesn't have to travel across membranes because basically the cells connect their cytoplasms directly. So it just allows for faster water transport. I have an electron micrograph of these plasmodesmata here, and you can see basically, like I said, you've got the cytoplasm being connected right between the cell walls. So that's the plant cell wall. Kind of the main thing to know is it's made of cellulose and that it's outside of the membrane. And some other definitions there, I might ask you what plasmodesmata are on the midterm, but it's not going to be like a big long answer question. It might be one multiple choice or something like that, or a matching question. So there are other cell walls, and I want to talk about and kind of just highlight what is important. So animal cells do not have a cell wall. We're going to talk about the extracellular matrix in a minute. Bacteria have cell walls. Most of them are made out of something called peptidoglycan. We've talked about that already. I told you I'd come back to it. Archaea do not have peptidoglycan or cellulose, but they might have other materials. Come back to that in a moment. Fungal cell walls are made out of chitin, and protists, there's quite a bit of diversity there. So what are the most important ones? Plant cell walls are important, animal cells, bacteria, and fungal cells. The archaea and protists, I've kind of just mentioned them for completion, and I think I have a slide on each of them, but really not going to go into any detail on those particular types of cell walls. So let's talk about the bacterial cell wall. The bacterial cell wall is made out of peptidoglycan. So I mentioned peptidoglycan way back in topic 2, and if you take a look at the word, you have peptidoglycan. So glycan, of course, means carbohydrate. That says carb, sorry for my bad writing. And peptido means we have amino acids. So if you take a look at the structure here, these diagrams here are called chair diagrams, by the way, and they're trying to do a little bit better of a job representing carbohydrates in three dimensions. And you have 2 carbohydrates here. We have NAG, which is N-acetylglucosamine, and we have NAM, which is N-acetylmuramic acid. So you don't need to know NAG and NAM, but they are basically glucose, and it's glucose with extra functional groups. So N-acetyl is like this little group here, and so that's a nitrogen attached to an acetyl group, and that's where the name comes from, N-acetylglucosamine, right? N-acetylmuramic acid is very similar to N-acetylglucosamine, except for it has this thing here. And this thing here is a bunch of amino acids. So alanine, glutamic acid, what's that other one? Pymalic acid, alanine. And so it's very similar to cellulose, but it has all these extra functional groups. Not really sure why it has all these extra functional groups, but it does. It's working for the bacteria and gives it a unique cellular structure. So one thing about this is the way it's structured is you've got NAG, NAM, NAG, NAM. So each of these here, maybe I should label them. We've got NAG here and NAM here and so on. You take these carbohydrates and you stack them together just like cellulose. You've got a whole bunch of hydrogen bonds, just like cellulose. But these amino acids, you can see right here, they link the strands together. So rather than having just hydrogen bonds holding the strands together. We actually have a whole bunch of covalent bonds. So it turns out that the bacterial cell wall is a very, very tough chemical structure. And you can do all sorts of things to it that will kill the cells, but the peptidoglycan persists. It's very, very tough. So I want to talk about cell walls, and it turns out that bacterial cells in general, there's actually two types of cell walls. And we call these things the gram positives and the gram negatives. So take a look at these structures. The gram positives, I'll tell you why they're called gram in a second, but they're named after a guy named Hans Christian Gram, who was staining these things. But take a look at the gram positive. The gram positive on the left, you have something very similar to a plant cell wall. And you have a membrane and you have the carbohydrate on the outside, so the peptidoglycan. The gram negatives, on the other hand, are more like a sandwich membrane and with a cell wall in there. So you actually have two membranes. So you have the plasma membrane here, and then we have something called the outer membrane. And in the middle is the peptidoglycan. So a little bit of a different system here. Extra membrane gives them a little bit something else going on in terms of their physiology. And it's kind of worth talking a little bit about the gram-negative cell walls, and I'll come back to them in a minute. I think I have some pictures here for you of the cell walls. I do. Here we go. So here are some pictures. This was taken actually by a friend of mine years ago. He was a very good electron microscopist. And you can see the gram-positive here on the left has a thick layer of peptidoglycan. You can actually make out the phospholipid bilayer right there. It's actually a very, very nice image. And the gram-negative on the right, you can see two bilayers and the peptidoglycan right in the middle. So very, very nice pictures. So let's talk about why they're called gram-positive and gram-negative. I mentioned that this gram is actually named after a person who studied these about 150 years ago, actually now, quite some time. And he discovered there were two types of bacterial cell walls using a staining procedure. So in the gram stain, he was using something called crystal violet. So crystal violet of course is purple. The gram stain works by one of the stages you're adding this crystal violet stain, and the crystal violet stain actually gets stuck in the thick layer of the gram-positive cell wall. And so they kind of end up purplish. The gram-negatives, they will retain some of the purple, but not very much. The purple stain does not really get into the peptidoglycan layer very effectively and can actually be washed away relatively easily. So with the gram-negatives, we use a second stain called saffron and red, which is kind of a reddish pinkish color. And so the gram-negatives turn out kind of a pinkish reddish color, depending on how much stain you've used in the procedure. So here are some pictures. You can see very different in terms of colors. The gram-positive stain of purple. And we actually talked about anthrax already. That's Bacillus anthracis on the left. And the gram-negatives turn out to be a pink or a reddish color. And you can see there's E. coli on the right, staining the pinkish reddish color. So we'll talk a little bit more about the gram stains next week in the lab and also in the lectures next week as well. Or actually probably this week, that's what I meant to say. There's a gram stain that I did, by the way, and you can see the two different types of species there, the gram-positive and the gram-negatives. Sometimes it's hard to get a really sharp picture of things this small, but I was pretty happy with this particular photograph. So more on gram stains in lab 4, that would be this Thursday, and topic 5, and we're going to
I thought there was a holiday this Thursday.
Oh, right. Sorry. Thank you very much for reminding me. It's already that week, isn't it?
Yep.
Yeah. No, so I guess that would be next Thursday. So thanks for the reminder there. So one other note to think about these gram-negatives, I just want to mention they have, like I said, they have this kind of a sandwich layer. And it turns out that what this means is that between those membranes, getting ahead of myself, I want to talk about periplasm first. Between these membranes, there's this, this little zone here. And it's kind of like a very, very thin compartment. And that actually has a name. It's called the periplasm. And it turns out the periplasm has enzymes in there. And some of those enzymes can give these organisms resistance to drugs such as antibiotics. So it turns out that gram-negative organisms historically have been a little bit harder to treat because they're a little bit more resistant to drugs. So there's some relevance there in terms of what's going on there. The other thing to point out, and I'm not going to get into really any detail in this class, but if you take any medical classes, is that the outer membrane actually has a unique lipid in there. It's called LPS, which stands for lipopolysaccharide. And this here is something unique to bacteria. And so it turns out it's an endotoxin. What does that mean? It means that if you get gram-negative organisms in your blood, so you get a blood infection, and these lipids, these LPS molecules are released into the blood, your immune system can get a little bit excited and can cause, in some cases, a very severe immune reaction. It's called septic shock. So I'm not going to get into any detail on that, but you can always ask me about it later if you're interested. Okay, so that's it for the bacterial cell wall for today. A little bit more on it in topic 5, a little bit more on it in lab 4, which will be next week. Thanks for the correction, Devin. What about archaea? These archaea are not bacteria, remember, they are their own domain as a part of the phylogenetic tree of life, and they do not have peptidoglycan. Now, some of them have a carbohydrate called pseudopeptidoglycan. So what is that? You can look it up. I can show it to you. It's a different carbohydrate. If you took a quick glance, it looks like peptidoglycan. But when you take a better look, it turns out that the glycosidic linkages are totally different, and which makes it an entirely different carbohydrate. So they have a unique carbohydrate. No surprise, they are a unique domain. No surprise that they have a unique carbohydrate in their cell walls. Some archaeal cells also have these things called S layers. S layers are these crystalline proteins, and you can see there's an electron micrograph image of what it kind of looks like. So anyway, something different because they are archaea. And I'm going to come back and talk about archaea in a few minutes when we start topic 5. So what about fungi? So fungi have chitin. Other than that, they're actually kind of similar to a plant cell wall. You've got a membrane and you've got chitin on the outside instead of cellulose. They have proteins in there. They're like pectins that are kind of sticky. Chitin is very similar to cellulose. It's just made of N-acetylglucosamine instead of being made out of just glucose. All right. Lastly, but not least, are the protists. And what can we say about the protists? The protists are a unique group, huge variety. Some of them have cellulose, they're kind of plant-like. Some of them have chitin, they're kind of fungi-like. Some of them have things that you don't see anywhere else. You can see a whole list there. Agar, algin, some of them have crystalline compounds made of calcium carbonate, and you can see that in these diatoms that I have down there below. Some very interesting cell walls, and some have no cell walls at all. So they're protists, huge variety. That's kind of one of the big things we can say about protists is there's a huge variety in terms of what they have. All right, so I want to finish off this unit and talk about animal cells, which are hopefully you think important because we are animals. Animal cells do not have a cell wall, but sometimes our cells have something called an extracellular matrix. So an extracellular matrix on first glance may sound like a cell wall. It's found outside the cell membrane. Sometimes it's providing structural support very similar to a cell wall. Sometimes it's doing other things. And in the human body, one of the main extracellular matrices is, of course, made out of collagen. So I think we talked about collagen a little bit already. I'd mentioned it's in your skin. It's kind of bouncy and flexible. It's a rope-like structure. So this extracellular matrix really is what makes up a lot of your connective tissue, and it's a big part of your body. So it's helping cells to stick together, but you can also have cellular messages spread through collagen and other things. So extracellular matrix, I'm going to give you 3 examples. So one is connective tissue. And that, of course, is mostly collagen. There are other compounds and macromolecules in there. If you take a look at this diagram, you can see this fibronectin, proteoglycan. If you go to different parts of the bodies, like if you're looking at the tissues in your knees versus the tissues between your muscle cells, you have different types of makeups, but kind of the big thing that's common amongst all of them is collagen. Collagen is a big part of connective tissue. So what about number 2? Number 2 is bone. Is a type of extracellular matrix. How come this is not writing? Somehow my eraser appeared. So bone, I'm just going to draw you a little picture over here on the left. Bone, you kind of have these little cells. So there's a cell, and there's another cell, there's another cell, and there's a huge amount of separation between these cells. And in between, you have these minerals. There's all sorts of different calcium minerals. I can't even remember all the names of them. All over the place. And that's kind of like what bone is like. You just have these minerals and they're hard and a few cells kind of in between. And the last example I'll give you is something similar to bone, which is cartilage. Cartilage. Okay, I don't know how to spell cartilage today. I think there's a G in there. I feel like it could be an A there though. Cartilage? I have to look that one up. For some reason I can't spell anymore. I used to be able to spell. Something happened in my brain. I guess I'm getting old or something. Cartilage is similar to bone. It's not as hard, it's a lot more flexible, and the cells are actually closer together. So you have cartilage in your ears, you have it in the tip of your nose, in places like that. They're a little bit firm, but not bone firm. Okay, so kind of the last sort of footnote to this topic is that there are other cell surface structures. I'm not going to ask you about any of these on the test. You can see there's a few defined here. There's tight junctions, and they basically bind tissues together. There's channels and all sorts of things out there. And if you take any more cellular biology courses, you'll probably talk a little bit about these in some detail. So that brings us to the end of topic 4. We're going to do a Kahoot and talk a little bit about topic 5 today. I want to talk about archaea. So load up your app and we will, oh, what did I do with it? There we go. Got to find the Kahoot and I will get it up here for you right away. Okay, I guess I just got to make sure I share the right screen. There we go. Okay, here goes, loading up. There's the game pin for you, 266-6486. This one has 8 questions, I believe. It's a little bit longer than some of the other ones. All right, we're going to start right away. If you want to join in, the code here, 266-6486, will be at the bottom of the screen. Okay, let's do this. Number 1, for the type of membrane protein that is labeled. Okay, so the correct answer is most of the amino acids are hydrophobic. That is not a peripheral protein. This is in fact an integral protein because it is integrated into the membrane. So remember, the integral membrane proteins are in the hydrophobic part of the bilayer, and so they have hydrophobic amino acids. All right, question 2, true or false? Phagocytosis is a type of cellular drinking. False. So it is a type of cellular eating. Phage means to eat. Question 3, facilitated transport requires energy. False. So facilitated transport is a type of diffusion. And as you can see in the diagram here, it requires a protein. Sometimes it's these hinged ones, sometimes they're channel proteins, but it requires no energy. So that is false. All right, Tom is on a hot streak. Well done, Tom. Question 4, cholesterol. So the correct answer is regulates membrane fluidity. So cholesterol, there's the picture there, that is not a protein, that is a steroid. It's a type of lipid. And the correct answer is it regulates membrane fluidity. Question 5, the fungal cell wall is made out of cellulose. So the correct answer is false. It's not shown here, but this material is actually chitin, not cellulose. Remember, it's the plant cell wall that is made of cellulose. Question 6, true or false? A gram-positive cell wall has a thicker layer of peptidoglycan than a gram-negative cell wall. True. Okay, that's good. Some people were listening. That was today. Question 7. Which of the following is true regarding the extracellular matrix? Okay, well done. Collagen is a connective tissue and is an example of an extracellular matrix. Okay, one more question, I believe. True or false? Plant cells like to be in hypotonic solution. The answer is true. Remember, hypotonic is like pond water, which is like water, and we like to give our plants water. All right, let's see what the podium looks like. Bob as bronze. Mohammed, silver. And Tom, gold. Well done, Tom. Okay, so covering a lot of material, by the way. Maybe you've noticed, maybe you haven't. But we are a week and a half away from the midterm, or sorry, 2.5 weeks away from the midterm. And so you may want to think about starting to look at your notes and whatnot soonish. And we're going to start talking about the midterm a little bit more next week, and hopefully give you a chance to prepare for it well. But in the meantime, you do have some study questions and some quiz questions up on Moodle you can look at any time. So topic 5 and topic 6 are the last 2 topics we're going to cover before the midterm. Topic 5 is on prokaryotic cells. We're going to talk mostly about bacteria, but we're going to talk a little bit about archaea today, and what are these creatures, and you know, why are they different from bacteria and archaea? I think it's a very good question. So let's start off, and I'll show you some numbers on some different organisms here. So first number, 5,400. Believe it or not, that's about the approximate number of species of mammals on the planet. And probably nobody knows this, but about 1,000 of those species are actually bats. Isn't that crazy? About 20% of all mammals are bats. Who would know? 10,000. That's about the number of species of microorganisms, prokaryotes, that you find in a teaspoon of fertile soil. More organisms in that soil, more individuals than the number of people who have ever lived. So really, really incredible amount of microorganisms out there in prokaryotes. What's 4,000? This is about how many that we've actually characterized and named. That's a pretty big number. And We think there's maybe around 4 million different species. New ones are being discovered all the time. It's really incredible. So some other numbers for you, 30 to 40 trillion. That's the number of cells in the human body. 35 to 60 trillion, the number of bacterial cells in and on your body. So we probably have more cells that belong to the bacteria than belong to us as a part of our body. These numbers are huge. So we're doing some important things, and we're going to talk a little bit about health and disease. That'll be next time, or maybe on Friday, depending on how far we get. Today, I want to kind of talk a little bit about these organisms, kind of in a general biological sense. So what are prokaryotes? Prokaryotes are, of course, organisms that contain no nucleus, and they contain the two domains, the bacteria and the archaea. And I want to talk about the archaea here in a moment. They are the most abundant and diverse organisms on the planet, and there's a lot of them out there. So interesting little facts for you. So kind of back to this slide. And what I want to do was basically make a little table and compare some characteristics of the eukaryotes and prokaryotes. So I think way back topic 1, I made some very brief notes regarding eukaryotes and the prokaryotes. So we're coming back to this, and I want to make some more detailed notes on this. So what I normally do is I normally do this on the whiteboard. But what I've done instead is I've started a Word document, and I'll just make some notes here, and I'm just going to type into the Word document. So you can see my heading is prokaryotes and eukaryotes, and then I have some kind of descriptors and characteristics on the left-hand side. So first, the prokaryotes, who does that include? That includes the bacteria and the archaea. So we already said that. The eukaryotes, this includes a lot of the organisms that we're familiar with, so animals, plants, fungi, those might be the obvious ones, and the one that we keep mentioning here, there are the protists. So protists, sometimes people might include algae in there as something distinct from the protists. Sometimes people group them in with the algae, or they group the two things together. I'll put it in brackets just for fun. The eukaryotes have a nucleus and the prokaryotes do not. Maybe we'll put that down. That should be the next one here. So membrane-bound nucleus, no. Membrane-bound nucleus, yes. So that's kind of the main thing. And the other one is size, which is something we talked about before. So prokaryotes' typical size is maybe 1 to 10 micrometers. So I'm going to actually insert the special symbol for micro. And it's right there, and that's a micrometer. So that is 0.001 to 0.010 millimeters if you need the conversion there for you. And that's 1,000th of a millimeter is a micrometer. Eukaryotes, maybe 20 on the small end to 1,000 micrometers is pretty common. Kind of depends. If you're a plant cell, you tend to be a little bit bigger than an animal cell. It depends on the tissue. You know, red blood cells tend to be pretty small. Our neurons in our body can actually be longer than 1,000 micrometers. That's 1 millimeter, of course. And so, you know, pretty big range there, but that's kind of a typical range. Okay, other membrane-bound organelles. No and yes. So that's another distinct thing, is that eukaryotes have all of those membrane-bound organelles that you learned about in high school. We're going to talk about in topic 6. So things like the Golgi body, things like lysosomes, endoplasmic reticulum. You don't find those things in the prokaryotes. Cell walls. We just talked about cell walls, so we can fill this in very easily. So remember that bacteria have peptidoglycan as their cell wall. Our archaea, so they have sometimes pseudopeptidoglycan. And some of them have something called S layers. What about eukaryotes? Animals. there is no cell wall, although sometimes they might have an extracellular matrix. I'm not going to write that part down. Plants, what do they have? They have cellulose as part of their cell wall. What about fungi? Fungi have chitin, and then we have the protists. And the protists, I'm just going to say very diverse, meaning that there are quite a few different options that they can have, many branches and many kingdoms in the protist part of the phylogenetic tree. And some of them don't have cell walls, some of them have, some of them are plant-like, fungi-like, and some are just very unique and don't look like anything at all. Okay, so I'm going to move down here. Maybe I'll shrink this just a little bit in case anybody missed anything. And now we want to talk a little bit about DNA. Okay, so what can we say about DNA? One thing I want to talk about is the chromosomes. So chromosomes in prokaryotes are usually, there's usually only one, and usually circular. So they're kind of, I don't want to say like hula hoops, they're more like elastic bands where they're a little bit more tangled up. What else can we say about the DNA? We have smaller genomes. And maybe I'll just leave it at that for now. We are going to talk a lot about DNA later on in the course. So what about eukaryotic chromosomes? Chromosomes. So kind of the opposite. Usually there's multiple. And eukaryotes, often you might have haploid or diploid organisms, or sometimes you have tetraploid, and there's a whole variety of ways you can do your chromosomes. Maybe not worth it getting to all that detail there. And they're usually linear, so they're not a molecule that doesn't wrap around on itself. We can also say they have larger genomes, often much, much larger. And one more thing that I want to say about the DNA is that the DNA is bound to histone proteins. And we're going to talk a little bit about histone proteins later on in the course, but these are proteins that are, they're basically there to package the DNA because there's so much of it, it just needs a little bit more help to be organized. We could also say that the DNA is found in a membrane-bound nucleus, so maybe I'll put that found in a membrane-bound nucleus, but we kind of already addressed that. Nucleus, there we go. Okay, so moving on. A couple more things to mention about prokaryotes and eukaryotes. Let's see here. What about the ribosomes? So the ribosomes in prokaryotes are smaller, and there's something called 70S. We haven't talked about what that means yet, so we'll get to there. And the eukaryotes have larger ribosomes. So different ribosomes, that they're different types of cells. Okay, moving down here, I guess that's on the next page. So what about reproduction? So you probably know that eukaryotes have mitosis and meiosis. So that's how the cell reproduce. And you can have asexual reproduction, and you can have sexual reproduction. A huge amount of variety in the eukaryotic domain. If you're an animal versus a plant versus a fungi, there's lots of things that can be done there. In prokaryotes, they don't do mitosis or meiosis. They do something that we call binary fission. So binary fission is a little more simpler, and we'll talk about that not today, but a little bit more next day. Reproduction is completely asexual, although maybe I will put a note that DNA exchange is possible. And it's through a process called conjugation. So I haven't talked about conjugation yet. We're going to get there. It's going to be next lecture. So some of these things are kind of just a little bit of a, I guess, an advertisement of what's to come. Okay, so I have one more column there, and you can see it says motility. So what is motility? Motility is, it basically means moving around or swimming or something like that. So bacteria, sometimes they have flagella, and the flagella that rotate. So maybe I meant to put rotating flagella. And not all bacteria have flagella, but that's kind of the common way to do it. And eukaryotes may have flagella as well, but these flagella are more whip-like. So they don't rotate, they kind of flop up and down. And they also have something called cilia. So more on all these structures as we get into further lectures. Also, some eukaryotes do something called amoeboid movement. And if we're lucky, we're going to actually observe some of this movement in lab 5 in person when we come back to campus. Okay, so hopefully everybody got all of those notes. If you missed something, let me know. You can either review the video, or you can always ask me and I can email you this.
Can you just go up a little bit?
I sure can, yeah. Yeah, if you ever miss something, just let me know. I don't usually post these documents, but it's pretty easy for me to email it to you if you want me to send it your way. Okay, so just looking at the time, I do want to talk about archaea for a minute in a bit of detail, and then I'll finish off today's lecture. So I'm going to switch back to the PowerPoint. Where's the PowerPoint? There it is. So back to this slide here. This was way back in topic 1. I mentioned we had 3 domains of life, and then I would talk about archaea. So let's talk about those archaea just for a minute or two. What can we say about the archaea? They were kind of discovered in these areas here. This is a hot spring, and people would put little microscope slides there and find things that look like bacteria, but the water is super hot in some of these things, like greater than 100 degrees. And it turns out there were organisms that lived in there. And in the modern era, when we had a better biochemical understanding, we realized these organisms were very different from the bacteria. Some of the archaea are not found in extreme environments, as you might get from this little cartoon. Some archaea actually are involved in making methane. And so that means they can live in your gut. So I wanted to make a couple of notes about archaea here. But one of the big notes to know is that they are found everywhere. There's no known pathogens. A lot less is known about them. So we're kind of mostly talking about bacteria when we talk about prokaryotes. So like I said, I wanted to write some notes about the archaea. So what can we say about the archaea? So first of all, remember that they are prokaryotes. And that, of course, means they have no nucleus. No nucleus. And also remember that they are a unique domain. That should have been my first note. Note 2. Remember they have unique cell walls. And remember that was pseudopeptidoglycan. Pseudopeptidoglycan. I know that's a big, long word. And we also had S layers. What else? We also talked about archaea as having unique phospholipids. Unique phospholipids. And remember that some of them are actually monolayers. So go back to the beginning of our lecture on membranes, and you'll see what I did mention it. What else can we say about these things? As I mentioned, there are no known pathogens. That means, as far as we know, none of them make us sick. And that means for studying these things, we have a lot less money available for research on them. They also tend to be pretty hard to grow in the lab. They grow very slowly and are difficult to culture. And so that kind of also makes another reason why we know quite a bit less about them. As I mentioned, they are found everywhere. And by everywhere, I mean everywhere living organisms live. And that some live in extreme environments. Some live in extreme environments. And I also mentioned that some are methanogens, meaning they make methane. So they're found in the guts of many types of organisms, and they're producing methane, unfortunately. The last note is that when you look at this particular group of organisms, there are many other unique type biochemical characteristics. I mean, I could list a whole bunch. They're unique in the way they make their RNA. They're unique in the way they have different proteins for the flagella and all sorts of other things. Not worth getting into other details here. We're not really going to talk about archaea for most of the rest of the course. I might mention them once or twice, but that's about it. So that's where I wanted to cover today, and it looks like we're out of time. So thanks for coming, and I will see you on Wednesday.